Hello everyone and welcome to the fifth in the lockdown webinar series. This one's on gut health. So this one's about the microbiome, and so we'll be looking at the bacteria that's in your gut and what can be done for helpful bacteria and what can be done for pathological bacteria. And when I say that, helpful bacteria produce things that are very helpful and meaningful for your microbiome, and the pathological bacteria produce stuff that's not good for you. So we'll be going through looking at the effects of the bacteria in your gut, looking at what's good and what's not good. And I'll be sharing with you some kinesiology formats that I've developed along the way that you can use in your clinic to help with the balancing of your microbiome. And this is really focused on the large intestine because, you know, the microbiome should be in your large intestine, and where you have really a lot of bacteria there. So intestinal bacteria, they do many things. They produce B vitamins, and we have transporters for all the B vitamins, which means we can absorb all of the B vitamins except for B12. It doesn't matter what people tell you, even if we synthesize B12 in your gut, you can't absorb it. We don't have the mechanism, the absorption of B12. It's a very specific and specialized mechanism that requires B12 with binding with the intrinsic factor that's in the stomach, and then it is absorbed through special receptors that also the same receptors that absorb hemoglobin. So we can produce the vitamin K, the short chain fatty acids, which are really important. Sometimes they're called volatile fatty acids, and you know, a long time ago, I had a friend who had a horse, and they wanted me to work on their horse, and so I looked up about horse nutrition, and when they have this horses have these massive cecums where they can digest the dietary fibre that we can't, and they produce a lot of short chain fatty acids. And I thought we didn't produce them, but I was shocked to learn that we produce them as well. We also they metabolize a lot of other things that are both good and bad, like bile acids, sterols, xenobiotics, and things like this. So our gut bacteria, in fact, are a really integral part of our body. And as you can see in the slide, we can have up to like 1 trillion bacteria in our intestine. And that can be up to, you know, maybe 300 to 1,000 species, but 99% are usually about 30 to 40 species, and these are the really important ones. And when you look at our fecal matter, 60% of the dry weight is the gut bacteria. And so we can really consider the gut bacteria like an organ because of their metabolic activities. Short chain fatty acids. So this is the interesting thing. What is it about short chain fatty acids and why are they important? Short chain fatty acids are also called volatile fatty acids, and they've got less than 6 carbon atoms. And usually when we talk about fatty acids in our body, we're talking about fatty acids with 18, 20, 22. They're the important ones in human metabolism, so that many carbons. So short chain fatty acids only have 6 or less, and they can cross the blood-brain barrier. And that's important because then they can have really, as we'll see, short-chain fatty acids are actually very helpful mechanisms, and especially in energy production, because in most cells, the normal cell can use either fatty acids or glucose to burn as fuel to enter the Krebs cycle, which produces electrons, which goes to the electron transport chain, which then, in the presence of oxygen, we can produce ATP, which is a unit of energy. So basically, short-chain fatty acids, because they're very small, they get broken down quite quickly rather than a long-chain fatty acid, like if you've got 18, 20, 22 carbons. Basically, it's like when we're breaking down fatty acids into energy, it's like slicing a salami. It's like the enzymes have got to like slice. They keep slicing them off until we get down to the acetyl coenzyme A, which can then enter the Krebs cycle, which then produces energy for us along the way. So looking at the short chain fatty acids, we have, by the number of carbons, you can see the very simple ones, formic acid. And, you know, then with 2, it's acetic acid, or the salt name's acetate. So that means basically there's just a slight difference in it. Then you've got propionic acid. Basically, it's oxidized and reduced forms of it. Butyric acid or butyrate. And one thing you should always remember, butyrate is your mate. It is one of the most helpful things you can produce. And if we have a lot of butyrate producing bacteria in our intestines, we will be very healthy. And I'll talk about that, how, you know, when we look at the different fuels for the gut, you know, like dietary fibre and resistant starch, you know, and especially the resistant starch, if we've got a lot of resistant starch, we will produce a lot of bacteria that produce butyrate, and that's your mate. Then you've got other ones, isobutyrate. It's just a slight variation. So it's got a chain, and valerate and isovalerate. But the important one is butyrate. And if you're getting your poop tested in a getting your microbiome, if you get your poop tested, you will have you will need to have if you've got a lot of butyrate producing bacteria, then you know you're in a healthy space. It's really one of the markers. Okay. So what's good about short-chain fatty acids? They're produced by the fermentation of fibre and resistant starch in the microbiome. They're absorbed directly into the portal vein of the large intestine. So that's basically the way you absorb fat. They affect the production of lipids, energy, and vitamin. They can be responsible for 10% of your body's energy. But this is what's important. 90% of the energy of the epithelial cells of the large intestine. If you want to have a healthy large intestine, you need butyrate. And this is not in this I just added in before, so you won't have this on your slide, but it'll be on the, if you're watching the YouTube video, it'll be there. I'll as usual, I will make the PDF available there, but you can see some of the things with the butyrate. It's a primary energy source for the colon. And so it's really important. If anyone's got any kind of colon disease, you need to support butyrate production, you know, like IBS, Crohn's, colon cancer, anything like that. Skeletal muscle, it increased lean body mass, and, you know, decreased fat accumulation. Adipose tissue, it leads to decreased adipose tissue mass and everything like this. And also in the liver, reduced inflammatory mediators, enhanced antioxidant systems. I mean, look at that. I mean, butyrate, that's why I say butyrate is your mate. Just remember that. And so working with the short chain fatty acids is actually quite important for that. Then you've got medium chain fatty acids. These have got 6 to 12 carbons. I'm not going to dwell too much on this, except when we look at the Hexa-Lipopolysaccharides later, which are a pathological product of the microbiome. They have 6 to 12 carbon atoms and help the process of excess calorie burning, weight loss, promote fat oxidation, and reduced food intake. So again, you know, like one thing is interesting about fat, you know, it's the one thing, you know, like even when you talk about the ketogenic diet, it's, you know, people say it's this or that, but it's actually a low, they say low carb healthy fat, but it's low the wrong carbs. It should be high the right carbs. And one thing, it's the biggest mistake people make on ketogenic diet is they end up eating too much protein. And they have not enough of the right carbs, which you need. And I'll be going through what is important. So these are the medium chain fatty acids. Again, once you start getting over the, you know, the low numbers, they're always in even numbers, and then up to, you know, 16, 18, you've got your linoleic and linolenic acid, which are essential fatty acids, and then up into icosapentaoic acid, arachidonic acid, which have 20, and then DPA and DHA have 22. They're the kind of important long chain fatty acids that we have a lot, and especially, you know, with fish oil and things like this, it's important to have good quality fat. And I spoke a lot about that in the lockdown webinar, chronic inflammation. And of course, for those people who did the chronic inflammation course as well. That was a couple of weeks ago. Resistant starch. Okay, so what is resistant starch? So there are 3 types of starch which affect the levels of blood glucose. You've got the rapidly digestive starch. So that's the not good. The slowly digested starch, that's the kind of, and then you've got the resistant starch, that's but of course, the rapidly digested starch and anything that's high sugar, like, you know, like fruit and everything, you know, when we lived in the jungle, our diet, fruit was seasonal. We might get it in the spring or the autumn. In the spring, we're hunting, we're mating, we're running around, we're doing all kinds of things. So we need a lot of extra energy. In the autumn, where we're going into winter, we actually needed to fatten up a bit, because in the winter there wasn't so much food, so we would burn down the fat. Even bears, for example, when just before they hibernate in the autumn, they go around and they eat all these fruits, so they put on all this fat for their hibernation where they're not eating, so they just burn the energy slowly over their hibernation period. And so, you know, this is the thing is that, of course, if you're doing like professional sports, running marathons or something like this, or doing really extra exercise, working out a lot, you need a little bit of extra easily digested starch, because you need that energy. You're going to burn it. It's just that in today's society, what we're doing is we're creating a space where we're eating all this kind of easily digested starch and sugars, fruits and everything all year round, but we're not burning it. So it just goes to fat, and we just put it on, and we get obesity. And especially with fruit, you get lots of fructose. I mean, that's just a huge problem. We have too much fructose. Glucose, at least, will create an insulin response, and we will start trying to use the glucose. We can either store it in the liver as glycogen or put it in cells through the insulin-dependent glucose transporter to use as energy production. And our brain uses glucose for energy, but they have non-insulin dependent glucose transporters, so they'll just absorb it anyway, because the neurons just use glucose for energy. So the resistant starch is the interesting one, and that just completely escapes digestion in the small intestine, and that's what it means. It's resistant to digestion. And it's the resistant starch is the important starch. And it's then, but then it gets down to your large intestine, and it produces, if you've got a lot of resistant starch, you will have high levels of butyrate, because the butyrate producing bacteria actually, what they do is they convert the resistant starch into butyrate. And if you don't have high levels of resistant starch, your butyrate levels start dropping, because that feeds those bacteria. They see the resistant starch and they start munching it, and they do all their thing and they produce all this butyrate, and that helps you. Okay. And it's very similar to dietary fibre. So it's a mild laxative, and it can cause flatulence. So what is starch anyway? I've kind of always thought, oh, it's chains of glucose, but it's actually got two components. One is amylose and the other is amylopectin. And so amylose is just a straight chain of glucose molecules like that. It's just kind of simple. Where amylopectin is the amylopectin, you see it's branched, and it's like, and it makes it more water soluble. So the amylose is linear polysaccharide. It's tightly packed, making it more resistant to digestion, and it's an important form of resistant starch. The amylopectin is branched like this. You can see it's got, oops, wrong way. You can see it's got branches of, what it means is branched is this is a branch of it. If it was just this would be amylose, but amylopectin's like that. You've got the glucose molecules branching off. And so this makes it more space. It makes it water soluble. It's easily digested. Branching takes place every 24 to 30 glucose molecules. Similar structure to glycogen. So glycogen is animal starch, basically. We store glucose in our liver as glycogen, so we reconstruct it. It's similar, but glycogen occurs every 12 glucose molecules. So what that means is that amylopectin is a plant-based form of starch, and glycogen's an animal-based form of starch. So if you ate liver, you would get glycogen, and again, it's branching. It's even more branched than amylopectin, so it's probably more water soluble and therefore more not resistant, and it would be more the easily digested starch. So here's the thing about resistant starch. So plants store starch in tightly packed granules, and you have layers of amylose and amylopectin, and see different plants have different if you look at the starch content, even of different grains, which have a lot of starch, the ratios of amylose to amylopectin is different. And the amylose is the good one. Amylopectin is the easily digestive one. So that's going to have a higher glycemic index. And so it's going to have a higher, it's going to raise your blood sugar levels. It's going to create an insulin response and everything like this. Whereas the resistant starch, you will get no glucose in your blood, no insulin response, no glycemic index. And so different plants have different size starch granules. The smaller, the easier to digest, and raw starch resists digestion due to the structure of the granule. It's the cooking. It's actually, so it's kind of interesting. It's, and there's a couple of things you can buy that if you want to support your resistant starch. One is psyllium. Psyllium husks is that wonderful ancient remedy from India. I remember living in India when I had dysentery, and I learned about psyllium then, because they knew all about it as part of their, it's native there. And also green banana flour. Green banana flour is full of resistant starch, or even green bananas. And just recently, you know, I'm from South American heritage, and so I love my patacones, which for those of you, most of you probably don't know what a patacon is, but it's a thing made out of green bananas. They do the cooking bananas, a plantain, and they squish them down and they cook them and they feed them to you. And when I bought some, there's a grocer here in Brisbane called the International Grocer, and when I go by there, I get some, and because they bring it from South America, I can't get it otherwise. And you know, and I looked at the sugar content is very low, and so if you want to judge when you look at any food, you look at the level, the number of carbs. You know, usually they have per 100 grams, and I always do that. You look at the total carbs and the sugars. The sugars should be really low, you know, and the total carbs much higher. When you see that, you know, when you've got low sugars, higher carbs, you know you've got good levels of resistant starch in there, because it's the sugars that are the ones that actually will create the problem. So the raw starch resists digestion due to the structure of the granule, and so that's really important. And you know, that's kind of interesting when you cook it. Water is absorbed. You know, when you cook your oats and everything's like shh, that actually changes it from resistant to easily digested. And so you burst the granules, and the amylose and the amylopectin all come out. And this is where you get, you know, like when you cook your porridge and it gets all that kind of gelatinous kind of structure. That's because you've burst your granules, okay? And you've made it more easily digested. So bummer about those of you who like your porridge, especially now we're in winter in Australia, but anyway. So resistant starch is kind of interesting, and so, you know me, anyone who knows me, like I like to know it all about it. But anyway, there are 4 types of resistant starch. There's the physically inaccessible or undigestible resistant starch, and this is what you get in seeds or legumes and unprocessed whole grains, you know. That's RS1. RS2 is inaccessible to enzymes due to starch conformation, as in green bananas and high amylose cornstarch. So there you go, something good from corn. Of course, not the high fructose corn syrup, that's not good. But the high amylose cornstarch, that's okay. So resistant starch is formed when starch-containing foods are cooked and cooled, such as pasta. So you know what? Having a cold pizza or cold pasta, that's not a bad thing. You know, there's nothing like having cold pizza for breakfast when you've got a hangover, right? It's one of my favourite breakfast items after, you know, there's the leftover pizza and had a few beers. It's like, oh, yeah, that's perfect. But then you can get up the RS4 or the starches that have been chemically modified to resist digestion. So, you know, they're the Frankenfood kind of things. Anyway, whatever. Dietary fibre. So fibre is important. So, so it is the portion of plant-derived food that cannot be completely broken down by human digestive enzymes. It's got 2 main components. You've got the soluble fibre that's easily fermented in the colon into gases and short-chain fatty acids. It delays gastric emptying, which can result in an extended feeling of fullness. Basically, what that means is that you eat less. So that's soluble fibre. Insoluble fibre resists the digestive enzymes in the upper gastrointestinal tract and provides bulk. So that that just gives you your bulk and makes your defecation easier because it absorbs water, and so that, you know, that yeah, it's basically anti-constipation, and so you get that, you know when you have a high fibre diet, all of a sudden you start feeling like you're pooping 3 times a day or something like this. It's like, oh my God, you know. It's interesting, scientists commenting rice is high GI, but if you cook it, freeze it, then eat it, reheated, lower. Exactly what I'm talking about. It's when you cook it and cool it, it seems to be better. So, you know, and because then what happens if you're eating the resistant starch, if you're trying to go for the ketogenic diet, you need the resistant starch because you need to feed your microbiome. And we're going to look at that. I mean, we've already seen that butyrate is your mate. But we'll have a look at if you have an unhealthy microbiome, what that means. So dietary fibre is made up of glucose, glucosamine, fructose, galactose, and xylose. Of course, those people who do NK will done the NK nutrition hologram series. You'll be salivating with all the formats. Here's some formats that I put together. Dietary fibre. So I use nutrition mode deep touch for diet. So like when we use nutrition like normal touch, that's what's in your, you know, when you use nutrition normal touch, that's what's going on in your like biochemistry. So I use nutrition deep touch, and then it's like, okay, it was in your diet. You put it in your mouth in your food. Anyone who's done my nutrition C course will know that. And then you've got supplement, which is should be nutrition light touch, spleen 16 on the right, by the way. So anything that's supplement, you know, you're taking a supplement, I use nutrition light touch. So you can diet, supplement, or biochemical. So any kind of nutritional format you have, you can triple them. And these are some that are important just so if you look here, the glycogen is nutrition times kidney 19 on the right. That's a biochemical because it's in your liver. But if it's dietary fibre or dietary starch, which is the same as glycogen, it's nutrition deep touch. And then if it's supplement, like you're taking a fibre as a supplement, you can get supplemental forms of it. You know, like if you're taking psyllium, like I did when I was living in India and I had dysentery and had diarrhea for 6 months. It was the only thing that would go through and not come out as water. And so then you would use nutrition light touch. So gives you the idea of how you can grade the different forms of the chemicals, whether it was dietary, supplemental, or it's a biochemical. Okay, so that's the story of starch, resistant starch, fibre, and butyrate. That is your mate. Next is IPA, and that is not a beer. It's indole propionic acid. And look at that, you can see that lovely structure that's typical of tryptophan. You all knew that, right? You can see the tryptophan hanging in there, derived from tryptophan. It's produced by specific human microbiome bacteria called Clostridium sporogenes. So Clostridium, and this is not Clostridium tetanus, which is bad. This is Clostridium sporogenes, and it produces IPA, and it's a potent neuroprotective antioxidant. So one thing, if you're getting your microbiome tested, is to get your IPA levels tested. You know, if you've got high levels of IPA, awesome. It decreases beta amyloid formation. OMG, WTF. That is what happens when people have Alzheimer's, right? They have high beta amyloid. So that means if you've got IPA, it's going to protect you against dementia and senile dementia and Alzheimer's. It decreases DNA damage. Huge. You know, most cells, you get damage to your DNA like 60,000 times a day. and your body repairs it. So, I mean, your cells work, your DNA is constantly getting damaged. And how is cancer formed? Errors and damage in DNA. That is the leading cause of cancer. It's derived from tryptophan. It's a potent scavenger of hydroxyl radicals, which that means it's an antioxidant. That they are a byproduct of just normal biochemical reactions. It lowers the risk of type 2 diabetes and associated with a high fibre diet. So let's look at some of the pathological things, and I'm not going to go through absolutely everything, but some of the important ones is Hexa-Lipopolysaccharides, trimethylamine, hydrogen sulphide, protein degrading enzymes, histamine, beta-glucuronidase, methane, ammonia, urease. That's kind of interesting. Tyramine, and other things, the B fragilis toxin, branch chain, amino acids, and GABA consumption. Do you know our microbiome makes a ton of GABA? And I know a lot of you kinesio nerds out there, we all know what GABA is, right? We know GABA is a really good neurotransmitter. It's inhibitory. It's one of the most common neurotransmitters in our brain. It's very calming. And it's also very important for brain function, that our brain functions really clearly. So we have certain pathological bacteria that produce enzymes that consume GABA. But having said that, there is no evidence that the GABA from your gut actually goes and is used in your brain. That doesn't mean it doesn't go there. It just means they don't know why we produce GABA in our microbiome. They don't know why, and they don't know what happens to it. I mean, GABA is very close to glutamate. It's a derivative of an amino acid. So something would happen to it. There's got to be a reason for it. But if we have our good bacteria, we produce a lot of GABA in our microbiome. So let's look at the Hexa-Lipopolysaccharides. That means you can see there's a lipopolysaccharide. Here's the polysaccharide bit up here, and here's the lipids. Hexa means there's 6 lipids. 1, 2, 3, 4, 5. You see all these little chainy bits there? And they're all, usually they're medium chain fatty acids, okay? So they have major functions. They anchored in the outer bacterial membrane, okay? So remember, they're on the bacteria and they provide protection for the bacteria. They are recognized by the innate immune system, which means, that means things like, monocytes and things like this, macrophages, anything that's gonna, like a phagocyte, really. And so they can initiate the clearance of the bacterial infection. And you can see the fat groups, these are saturated fats, so no double bonds. And they're 10 to 18 carbons, which these are the 10 to 18 carbons, what you see there. They're the saturated fats for 10 to 18 carbons. So what happens if you've got, hexaacylated, that means 6 lipid groups on the lipopolysaccharide. It's an agonist at this MD2 toll-like receptor 4 complex on the macrophage, upregulates inflammation. So those of you who watched the previous one on, I did on inflammation resolution, we know that when the macrophages are in that, like, if they're monocyte-derived macrophages or the M1 macrophage, they're upregulating inflammation. But when there's 4 or 5 acyl groups, so instead of having 6, you've got 4 or 5 hanging there. They're an antagonist at this receptor and therefore blocking inflammation. So what does that mean? Agonist and antagonist. An antagonist occupies a receptor but doesn't activate it. An agonist occupies a receptor but activates it. So when you've got antagonists and agonists at the same time, they compete for the receptor because only one can be there. So if you've got more of the 4 or 5, which would be the lipopolysaccharides and the tetralipopolysaccharides, they will compete with the Hexa-Lipopolysaccharides to occupy the receptor. If you've got more of the good ones, they'll spend more time at the receptor and therefore not activate inflammation. And here you can see, on the left, E. coli producing hexaacylated, and then you've got on the right this Sphaerides. It's producing pentaacylated lipopolysaccharide. So that will block it. And basically it looks like this. The hexaacylated and the here they call it underacylated, which is a 4 or 5, that'll block it. And this is the toll-like receptors, and these then if these are activated, then they trigger the cascade that will lead to inflammation. And you could refer, if you didn't see the previous talk on the chronic inflammation resolution, you can go on the YouTube channel and look that one up and hear me talk about that and the difference between the resolution and the activation of chronic inflammation. And inflammation should be there as an acute response, but if you've got high levels of the hexa-lipopolysaccharides, high levels of them, you will get chronic inflammation because they'll keep activating the monocytes into the inflammatory state, and they'll produce pro-inflammatory cytokines. In the M1, or will be the monocyte-derived macrophages, depending which one you wanted to look at. Anyway, so that's the hexa-lipopolysaccharides, and so then you've got the trimethylamine. The trimethylamine, you can see that it's got an amine group, which is nitrogen with 3 hydrogens, but here it's like got a nitrogen with 3 methyl groups. You could say that's an over-methylator. That's supposed to be a nerd joke. Anyway. So it's produced in the gut microbiome in humans. It's from phosphatidylcholine, betaine, choline, lysine, carnitine. It's oxidized into trimethyl N oxide, either by a microbiome or in the liver. And so this TMAO is associated with type 2 diabetes, cardiovascular disease, chronic kidney disease, increased inflammation, decreased bile synthesis, atherosclerosis, foam cell formation. That's not good. TMA is produced from carnitine, lysine by the following pathways. So you've got lysine, carnitine, TMA. There's all these enzymes. So that's not a good thing, right? Doesn't look good. We don't want cardiovascular disease, do we? So if you've got high levels of TMA in your microbiome, not good. Hydrogen sulphide, produced from cysteine, which has sulfur in it. It's protective at low energies as an energy source, dangerous at high levels because it inhibits the mitochondria, so it's energy production. In leaky gut can cause decreased gut motility, elevated levels in IBD and colon cancer. So, you know, if you've got the inflammatory bowel disease or cancer in the colon, often you've got high levels of it. It's not good. And also is responsible for stinky farts, which is not good. Ammonia. Well, this is something that really surprised me. So in the liver, we have the urea cycle, and we, you know, get ornithine, carnitine that goes around, and you produce urea. Then you think, oh yeah, my urea is going to go to the liver, and you know, blah blah blah. Then I'm going to pee, and then it's out. But 25% ends up in the colon. It's like, say what? And then you've got some pathogenic bacteria can produce the enzyme urease that actually produces ammonia from the urea. So basically you've done all this work to get rid of your ammonia, which is what the urea cycle does. It's like, because ammonia it's just an NH3. It's like a lone amine group floating around. And so you often get as a byproduct, especially of amino acid degradation. And so we've got this whole urea cycle to get rid of it. And then we pee it, you know, like it's, you know, urine. Actually, the word urine comes from urea. But then we can, if we've got the wrong bacteria in our gut, we actually recycle it into ammonia. So we're basically, you know, we're only getting rid of 75% of our ammonia. So you end up with high ammonia levels, which can lead to inflammatory bowel disease, chronic kidney disease, increased gut permeability, that's leaky gut, neurotoxins. Oh, that doesn't sound good, does it? Histamine. Some bacteria can produce histamine from the amino acid histidine. Histamine is involved with inflammation, IBS, food allergies, and asthma. Histamine is made up from the amino acid histidine. And so, and also histamine, it's known, we produce it a lot in food allergies, mast cells, and histamine degradation. And so it really is there. And this histamine that's released from mast cells is supposed to be a defense against parasites, but but if we're in the intestines, we're producing a lot of histamine from our microbiome. It's like, oh my God, inflammation city. And there you get all this inflammatory response. But what happens is, because it's not a natural inflammation, you know, usually when you've got the inflammatory response, you've got all your phagocytes and everything in there clamoring around your monocyte, your monocyte derived going in there to be converted into macrophages and everything. They're all like clamoring around to get in there, where there's no clamoring with that. And so then you increase the permeability, and whatever's there can get through. This is where you get, you know, all kinds of problems, especially with leaky gut and stuff like that. So methane, it's produced by specific enzymes, lowers gut motility, and it's associated with constipation and IBS. GABA consumption, as I mentioned before, GABA is, there's a number of bacterial enzymes that produce GABA in the microbiome. There are bacteria that will consume GABA as well. And so what's important is that, you know, we know GABA is good. It's what is not known is what's the fate of the GABA in our microbiome. But it is assumed that you've got to assume that we don't want to consume GABA in our microbiome. We would want it to leave and travel around our body. Interestingly enough, because you know me, anyone who knows me, I'm so OCD, I like to look up all the little nitty-gritty things, is that the bacterial enzymes that consume GABA, I actually looked them up. I could find them because I'd look at, there's a website where you can look up a substance and look at every known biochemical reaction, and so you can look up every enzyme, and I went through every single one, and I only found 2 that exist in bacteria, and that was those 2. Put enzyme numbers there for the NK kinesio geeks that have done Nutrition A and have got that whole complete manual of formats. Branch chain fatty acids, it's like leucine, isoleucine, and valine. When they're high levels in the gut, you've got a high protein, low fiber diet, which is not good. High protein, low fiber diets are really not good, and you end up with a lot of, as I'll mention in a minute, a lot of the protein degrading enzymes. And so high levels of the branched chain amino acids associated with obesity, type 2 diabetes, non-alcoholic fatty liver disease, which again is an obesity there, insulin resistance and decreased insulin sensitivity. They're all conditions of obesity or connected to obesity. So it's basically, if someone's got high levels of branched chain amino acids in their poop, if you're getting their poop tested, you would really want to look at, you'd really want to look at these obesity issues. They probably have issues with their weight and these other complications that are associated with it. Tyramine, this triggers the adrenal medulla to release noradrenaline above normal levels, and so noradrenaline is part of the sympathetic nervous system. What it does is it raises the blood pressure by triggering the renin-angiotensin pathway, which basically causes your adrenals to produce aldosterone, and that raises your blood pressure. So high levels of tyramine would generally cause people, they'd be in like a fight, flight, high blood pressure. You know, fight, flight, high blood pressure is like, I'm in danger. I got to run, or I got to fight to survive. I got to, you know, I got to lift this. I got to fight this to survive. So you get this high blood pressure to fight or to run, you know. But if you're not in that state and you're in that kind of high blood pressure state, that's not healthy. This is an interesting, you know, dietary fibre, and these are all the butyrate, propionates, again, another short chain fatty acid. If you look back on that slide, it's the one with 3. You can see these are all the enzymatic pathways for the production of butyrate and propionate, which are really good and helpful. And it was interesting when I was looking at this because, you know, when I did the put together the balance for this, like the really in-depth one with all the little details, what I found was there are bacterial enzymes that cross these pathways. So they draw it like this, but it's far more complicated than that. It's like unbelievably complicated. So you get little things here, little things there. This one's going this way, this one's going that way. But at the end of the day, what you want to know, have you got high levels of butyrate, propionate, IPA? That's what you want to have. And the other things like tyramine and everything else, you don't. So the balance. So we're going to be working, obviously we're going to be working in the large intestine. I mean, obviously if you've got SIBO, which is the small intestine bacterial overgrowth, that's another story. So here we want to balance the health. So large intestine, tensor fasciculata muscle test, bacteria mode. You've got these formats that I showed you before. This should be nutrition light touch there, supplemental fibre. That's a mistake. we're going to do our short chain fatty acids and use the nutrition times stomach and liver on the right. I showed you that in the previous one for the fat. You know, that's just generally a fatty acid format. Enzymes. So because we've got the enzymes to produce it from the bacteria, and explore the pathogenic factors, you know, go through those different ones and see LPS or tyramine or ammonia or histamine or all the TMAO, you know, all the things that are a problem. So as you can see, the different things you can do with the balancing the gut is that you can work with the microbiome in the gut and explore the good things, and understanding we want to do, you know, obviously modification of the diet, resistant starch and fibre is important. And I would encourage you, if you want to know more about that or you want to work with someone, is to go to a duly qualified health professional that's trained in that, you know, someone like an integrative doctor, nutritionist or a naturopath, something like this, where they can guide you in that. But, you know, like very easily, if you want to do it the easy way, you can go and get some green banana powder or some psyllium, and they will, and take that daily. That will really help you and feed it. You know, have lots of fiber in your diet, lots of vegetables, salad items. You know, the one of the problems with say the ketogenic diet, and certainly when I had my poop tested, because I do keto, well, I try to, is that I was almost good with everything except I had high levels of protein degrading enzymes, which meant I had too much protein in my diet, you know. But God, I love a nice big fat steak. Anyway, so that's hard for me to give up. But the thing is that it's about balancing the diet, and certainly by feeding the fiber and resistant starch is very important, and you can do that, you know. So and so anyway, so that's my talk for today, and I hope you all enjoyed it. I will be posting the YouTube later. So thank you very much.